So energy sources for the muscle contraction is the ATP is the major source. ATP stands for adenosine triphosphate, right? And you have these high energy molecules, right? These phosphates. And as they break apart, energy is released, right? So we see here ATP molecule, right, is broken down to ADP and the phosphate, and this is releasing energy, right? And this is needed to power processes, for example, as muscle contraction. Where do you get ATP from? So ATP is made from reuptake of nutrients. We eat carbohydrates, fat, glucose, right? And where, by the way, where's ATP made? Which organelle makes ATP in our bodies?
The mitochondria.
Yeah, mitochondria makes ATP, right, with oxidative phosphorylation, right? And this is what we essentially use. It doesn't just power muscle contraction; it powers a lot of processes in the body that need energy, right? So this is what I was talking to you about when it creates this power stroke, right? ATP needs to come in and needs to disassociate, right? Dislodge this myosin head, right? That's bound to the actin, the thin filament. ATP binds to it, dislodges it. Now it's cocked and ready for the next action. ATP is hydrolyzed into ADP and phosphate, right? Inorganic phosphate. And this energy, right, is what is utilized in order to facilitate the contraction of the muscle. Now, here are the processes that govern this, right? So we have breakup of glucose, which is essentially called glycolysis. This is the first part here. This, I have a question for you guys. There is, we just talked about, right? The mitochondria is what makes ATP. There is cells in your body that do not have mitochondria, at all. So if they do not have mitochondria, they can only do glycolysis, right? So they cannot go down here. Does anyone remember what those cells were that do not have mitochondria?
Red blood cells.
Right, red blood cells, excellent, right? So red blood cells do not have mitochondria, so they cannot do oxidative phosphorylation. They can only employ glycolysis, which is minimal amount of ATP. But our muscles, they need a lot of ATP. So that's why the pictures had mitochondria, right? And the neurons and the myofibrils there, you saw that, right? So glucose is broken down into pyruvate, and then this pyruvate enters into the citric acid cycle, happens in the mitochondria. And then the end results, right? When there is different intermediates that occur here, right? For your purposes, you don't need to know them. If you decide to learn biochemistry, you'll definitely memorize all of them. These intermediates then, right, have electron carriers, or I should say hydrogen ion carriers, as you see hydrogen ion here. They're carried by NAD or FAD. They're carried to this electron transport chain. And what happens is it creates this electromotive force where it pumps hydrogen ion, right, here. And there's this ATP synthase, right, that's found on the mitochondrial surface. It basically, for every hydrogen it pumps in, it like churns. As it churns, it creates ATP, right? The guy who basically discovered this ATP synthase, he won a Nobel Prize. I think his name was Mitchell. And for a long time, people thought he was crazy, describing this thing twisting in the mitochondria, bringing hydrogen ions in, making ATP, right? But that's actually how it works. You make this ATP, and then this ATP can be used for the functions that require energy, right? So now, any of you guys go to the gym, work out, and you like the best one I can do is you are working out and you're doing, let's say, like this burnout set, and you're doing the set until you can no longer do it. Any of you done that on a machine or free weight?
Yes, sir.
Yeah. So when you do it, you feel this burn, right? And what is this burn? This burn is basically this lactic acid. You start to make lactic acid. Why? Because as you're performing the exercise, right? It's as the time passes by, it depletes all your oxygen stores, right? It goes from aerobic to anaerobic, and the more you do it, the more lactate you make. That's why you have that burn, right? Like lactic acid is produced, right? This lactate can go into the liver. The liver can utilize it, right? To make glucose or other functions, right? So this what happens in a state when oxygen is insufficient. The other time when this occurs maybe you guys heard of this condition called, sepsis, right? In septic state, the tissues in the body, they have lack of oxygen, right? Because tissues are high metabolic need at that point. They require all the oxygen it can get, but it cannot get it. So tissues then transition into anaerobic, right? Glycolysis, start to make lactate, right? That's why we measure in the hospital, we check their blood level for lactic acid, lactate. If lactate is elevated, we know that their body is deprived of oxygen. It's making lactic acid. And that's one of the labs you could look at if the sepsis is improving, right? So in the anaerobic condition or in cells that do not have mitochondria, right, you can only make lactate or stick with glycolysis only, right? But in oxygen sufficient areas, right, the pyruvate can go into the mitochondria, and with oxygen in the electron transport chain, it can make a lot of ATP. So here we see oxygen is available, right? And so ATP is produced, right? The other thing I want to say for the storage purposes, let's say glycogen, right? Glycogen, the major source of it is in the liver, right? And we also have glucose stores in the muscle. But the biggest problem in the muscle is that the muscle, to go from glucose 6-phosphate, right, and then to break this down into glucose, it's missing a key enzyme. So muscles don't have this enzyme. The enzyme is being glucose 6-phosphatase. So muscles cannot readily give you glucose. So they cannot just break down glycogen and bring glucose into the blood. So due to that, right, liver is the predominant organ to do that. So liver can break down glycogen into glucose, but muscles cannot. And that's why muscles go into this lactic acid form. They make lactate, right? They cannot take glycogen and make glucose. So this is basically shows you muscle, right? It does store glycogen. It does go up to glucose 6-phosphate, but from glucose 6-phosphate, it cannot make glucose. It cannot. Why? Because it's missing that key enzyme, glucose 6-phosphatase, right? It makes pyruvate. Pyruvate can be further be made into lactate by lactate dehydrogenase enzyme. Pyruvate can, as it's made, right? Pyruvate has two fates. It can come out as pyruvate and go into acetylcholine in mitochondria. Lactate can be used in the liver, right, for that functions. So this is just another schematic, right, to show you the breakdown and what you want to remember, right? The muscle is missing glucose 6-phosphatase enzyme, so it cannot, break that into glucose. You see how it says glucose 6-phosphatase only in the liver? So liver can take glucose and make it available in the blood, but muscle cannot do that, right? You see how it says muscle and cannot do it. So it takes pyruvate, makes lactate, right? And the breakdown of metabolic metabolism here, CO2 and H2O, right? So that's very important. This here is when we take glucose, right? How much net ATP are you expected to make? So if in glycolysis, right, for every glucose molecule, you make about 8 ATP, but in mitochondria, right, if oxygen is available, right, you make about 38, right? But if oxygen is not available, you make total of 2, right? So here, as you can see, right, in the anaerobic conditions, you make less ATP, so cells will be deprived of ATP, and you will not get the muscle contraction that's needed or other processes that require ATP. So aerobic respiration is required. That's why in the cases of shock, when patient is saturating low, why do we provide them with oxygen? When low that hemoglobin, we want to make sure the oxygen reaches the target tissues, so those tissues are not deprived of oxygen. We also want to make sure ATP is done. Why? Because ATP is not only used for muscle contraction, the cells are used for other processes, sodium, potassium pumps, sodium, potassium ATPases, and other functions, right? So here we see, right? Why the other thing is why red blood cells only have glycolysis, they don't have citric acid, is because red blood cells action or the activity, they don't need as much energy, right? They don't need as much energy as a muscle would. Their job is basically just to transport, oxygen, carbon dioxide, right? And diffusion. So for their energy needs, for their metabolism, they only need lower amount of energy, so glycolysis is sufficient. But for all other cells, right, we need the citric acid cycle. We need aerobic conditions for that to occur, right? So here we see, right, as I just said, muscle fiber cannot, in cases where body requires glucose, it cannot just take its glycogen stores and make, it cannot make glucose. So we see glucose coming in, it does come into glycogen stores if it's excessive glucose, but in the muscle, because it's missing that glucose 6 phosphatase enzyme, it cannot basically take glucose and bring it into the blood. The only thing it can do is it can make it to go into lactate form, and then lactate form can go into the liver, and liver does have that enzyme, right? It can convert lactate into pyruvate, lactate dehydrogenase, and then pyruvate can be turned into glucose. So liver has all the enzymes necessary. Muscle does not. And muscle is kind of selfish. It keeps its oxygen stores with myoglobin until it's absolutely necessary, and it keeps the glycogen for its own uses as glycogen for metabolic needs like work, exercising, walking, activities of daily life, right? So muscle is selfish in that regard, right? This is just some of the disease processes. This I'm gonna leave for the neuro chapter, as well as tetanus. Tetanus is basically suppression of inhibition. Someone asked me, will you be in with succinylcholine? Will you be in a prolonged tetanus? No, you won't, right? You'll be in flaccid paralysis. Tetanus is suppression of inhibition, right? Tetanus is maybe you saw the pictures where the guy's heavily, severely contracted. That does not occur in succinylcholine. What we focused on today, right, was on this portion here because we're talking about the muscle. We talked about botulism, right? What does botulism do? Can anyone tell me?
Botulism, it's the one that blocks the receptor, the acetylcholine. Right?
Which specific mechanism it blocks? You want to be specific.
The acetylcholine attaching to the receptor to.
Yeah. So
No.
Yeah, so you're not right track. So botulism works on that synaptobravin syntaxin, does not allow the vesicle to bind, right, to the membrane. And what about myasthenia gravis? How's that work?
That's the one that with the nicotinic cholinergic receptor.
And what's specific about it?
Does it block those receptors?
The antibody mediated, antibody mediated. Antibodies that come in, they bind to those receptors, not allowing acetylcholine to bind. So that's what you want to keep in mind for this purpose. This is just some of the pharmacology stuff that I want to show you, right? This was succinylcholine in emergency medicine. So where does this occur? It's on the neuromuscular junction specifically, right? And what it does is it does not allow, right, acetylcholine to bind there, right? So what happens is initially you have fasciculations, as I said, right? And then you have that prolonged paralysis that occurs, right? This is the actual neuromuscular junction. This is the receptor, right? We see here in the myasthenia gravis patient, right? As I said, these receptors is what being affected. For acetylcholine, for succinylcholine, they bind here specifically. First depolarize the end plate, and then, right, give you that flaccid paralysis. Acetylcholine esterase, that's the enzyme that's found in that cleft, which cuts up acetylcholine. Right. This is a close-up view, right? If you guys wanted to see, these are the nicotinic cholinergic receptors or acetylcholine receptors. They have different domains, right? They have 2 alpha subunits, beta, gamma, and delta, right? Alpha, beta, gamma, and 2 alpha. When acetylcholine comes in, right, this is where it binds. So it binds here. The moment it bound, right, we see that calcium can come in, right? Sodium can come in. Sodium comes in to depolarize. cells are bags of potassium. Potassium leaves, right? And calcium was found on the outside. That's why calcium can't come in. What's the purpose of calcium? Calcium is an ion that causes exocytosis of the neurotransmitter. So calcium causes the binding of the vesicle and release of acetylcholine. And calcium also causes the contraction, right, to occur in the muscle. It binds to the troponin, and this will move the trypomyosin away, allowing the myosin head bind to the actin filament. So this is how you see how it actually exerts its effects here, right? This was the drug therapy I was talking to you about, right? So look, this is the molecule of acetylcholine. This is the normal molecule. And here we have this drug, the succinylcholine, right? It's kind of like the two acetylcholine molecules combined together. See, that's why it exerts those effects, right? It doesn't dissociate. And initially, it's kind of like acetylcholine. It'll bind here, right? It'll bind to this receptor, and sodium will come in, and you see this depolarization. But then it will not unbind. It will not unbind, causing that flaccid paralysis to occur. We also talk about rocuronium, pancuronium, right? These are non-depolarizing agents. So this is depolarizing agent. These are non-depolarizing. They act like that tuba curare drug. They basically come to the receptor here, and what they do is they kind of like cap it. They basically don't allow acetylcholine to bind. If acetylcholine doesn't bind, right, you cannot depolarize because no sodium is coming in, potassium is not leaving, right? So you're basically just blocking that site. But depolarizing agent, you see the fasciculation is occurring. But these are just different drugs, right? Rocuronium. Some in the hospital they'll call it rock. They'll call this SUX, right? Just abbreviation, VAC. Right? This is the medication, right? This is the succinylcholine that I was trying to explain to you. So it tells you, right? You must have immediate artificial respiration and oxygen available if you're going to use it. You definitely want to be able to ventilate. In New York City, you do not have this medication, right? In New York City, you're not allowed to administer this under REMAC protocols. Where does it bind? It binds here, right? And it does not. It first depolarizes the cell and then doesn't unbind, right? So here the specific action on the effect of it, the depolarizing agent like SUX, right? They bind to this alpha subunit specifically, right? The alpha unit here, and same way like acetylcholine will do, right? And once it binds, it opens the ion channels, and sodium does come in, contraction is made, and you see this as a fasciculation, the twitching, right? And these drugs are not broken down by acetylcholinesterase, right? They are not removed. So they stay bound to that alpha subunit for a much longer period of time, and they cause this persistent depolarization, right? So you'll have this neuromuscular block, you have flaccid paralysis, the patient cannot breathe. If you're not breathing for them, they will die, right? The non-depolarizing agent, we talked about the, rocuronium, right? Pancuronium, bancuronium. They compete with acetylcholine, right? So if they compete with that acetylcholine, whichever one binds or is in the excess, it will overcompete the other. So if I have a lot more of this non-depolarizing drug, it will compete with acetylcholine and it will bind here, right? And it will not cause depolarization. So it's blocking it. Now, if I want to overcome this, I can give them acetylcholine esterase inhibitor drug, right? If I give acetylcholine inhibitor drug, acetylcholine esterase inhibitor will stop acetylcholine esterase from working. More acetylcholine will be there. Now I have more acetylcholine in proportion to this non-depolarizing drug. So maybe I will overcome this paralysis so I could reverse it, right? So here we see, right? It binds to the alpha subunit channel, and the receptor will not open and no current will flow. So if no current flows, from that we can deduct there's no depolarization occurring, right? And the muscle will become flaccid. Right? So that's basically it. So what they say here is SUX is the only neuromuscular depolarizing drug that we have that's currently used. It is in fact used. You will see this in your ER rotations for a procedure known as RSI, rapid sequence induction, right? Which you probably read in your respiratory chapters, right? So we talked about that. This is your New York City REMAC protocols. The reason why I have it here, I have some problem set questions that I'm going to ask you to give me explanation about succinylcholine. So here, these are your medications. Out of all these medications, we do not have any neuromuscular blocking agents, right? So it's very important for you to know that if you have a patient who has, let's say, masseter spasm, they have laryngospasm, right? The larynx is spasming. You cannot break that with your drugs because none of them are neuromuscular blocking agents. Okay. We talked about the myasthenia gravis, right? It's basically a Greek definition. So it means it's heavy muscle weakness, right? So Greek, weak muscle, gravis means heavy. It may not be severe. Why? Because if the patient just, started having those signs and symptoms, initially it may not be such severe, right? They will have some weakness, but they might not be severe. So probably a more appropriate name would be heavy muscle weakness for these patients, right? This is myasthenia gravis mechanism, how it works, right? So here we have this receptors. We have the acetylcholine that came here. This is nicotinic acetylcholine receptor, right? This is normal physiology. So acetylcholine comes in, it binds, sodium comes in, depolarization occurs, right? And we have muscle contraction. Now, in myasthenia gravis, we have this anti-nicotinic cholinergic receptor antibodies. These antibodies, they bind here, and they don't allow, right, acetylcholine to bind. And if acetylcholine cannot bind, you don't have response. Also, they are degraded by acetylcholine esterase enzyme that's found here. So it's this enzyme that's found here, they're degrading acetylcholine, plus these antibodies not allowing acetylcholine to come in, muscle weakness, right? So how do we combat that? We give a drug called acetylcholine inhibitor. Acetylcholine inhibitor blocks this. So more acetylcholine neurotransmitter is available. Hopefully it will bound to those sites not occupied by antibodies. So you have increased response. This is basically from New England Journal of Medicine article. They basically show you the mechanism of injury, the mechanism of action, right, of acetylcholine esterase, right, and how the antibodies are working to basically inhibit the response. And we have drugs, right, to allow better inhibition of that enzyme, more acetylcholine to work, right, and better neuromuscular transmission, right? We talked about the botulinum toxin, right? Actually, besides this, right, you may have heard of Botox injections, right? They give you Botox injections, so all your skin, right, wrinkles will essentially dissipate, and they will be, stretched out. What is a Botox injection, right? It's from this bacteria, Clostridium botulinum. It makes this anaerobic gram-positive toxin. And how does that work, right? The way it works is we just talked about, right? It comes in here, right? So let's talk about normal, right? Normal, you have acetylcholine in this vesicle. Calcium comes in, right? Calcium comes in. The synaptobravin comes and binds to syntaxin. Once they are bound, the acetylcholine is released, and you have normal depolarization taking place. Now, somebody who got Botox injection, or they, consumed a canned good that was expired, it had a lot of botulinum toxin in it, right? So what it does, it comes in and it cleaves, it cuts up all these snare proteins, the syntaxins and synaptobravins. If it cuts these proteins, we can see it cannot bind, right? It cannot bind, it cannot release its contents. So the way botulinum works is it essentially cuts up these synaptobravin and syntaxin with the proteolytic enzymes that it houses, right? Not allowing to come in. The other thing we talked about, right, is tubercularity with the South American people. They use this plant. They basically would coat the arrows of, and use it to hunt their prey. It hits the prey, paralyzes, right? How does it work? Blocks the acetylcholine receptors. There's no depolarization. There's flaccid paralysis, right? Similar action to like rocuronium, pancuronium drugs,